Okay, thank you very much. As you know, my name is Kyriaki-Evangelia Aslani, and I am a postdoctoral researcher at the University of the Peloponnese in Patras, Greece. And also I am the moderator for today's session. I will present you my research entitled, sorry, just to move this here. From physics to bioengineering, the effect of micromagnetorotation on blood flows under strong magnetic fields. So my presentation will briefly cover the following. First, I will do a short introduction to what micropolar fluids, ferrofluids, and blood flows are, how they're related to micromagnetorotation. Then, we'll talk about the motivation and the objectives of my research, as well as clinical applications. Then, we will see the mathematical model that is used for describing all these phenomena. After that, we will see some first results of analytical studies on simple two-dimensional blood flows. And then we will see some numerical results on more complex three-dimensional flows. Finally, we will discuss the conclusions of the research, as well as some possible future directions. So what are micropolar fluids? They are fluids that contain small solid particles suspended in the mother liquid, each one possessing its own internal rotation, or spin, as we can call it. They were first modeled by Engen in Princeton in 1964. So key features of the micropolar fluid theory include, first, the microrotation, abbreviated as omega, which refers to the average internal spin of the particles. It must be solved alongside the fluid's velocity and must differ in magnitude from the fluid's vorticity, which is abbreviated as capital omega, in order for the micropolar fluid theory to hold. In micropolar fluid theory, we also have the introduction of an antisymmetric shear stress tensor, which is a function of the difference between microrotation and vorticity. And we also have the introduction of two additional viscosities, apart from the classical dynamical viscosity mu that we know from the Newtonian fluids, namely the rotational viscosity μr and the spin viscosity gamma. Typical examples of micropolar fluids include the liquid crystals, certain lubricants, and of course, blood. So what about ferrofluids? First, ferrofluids can be modeled as micropolar fluids because they consist of a liquid that contains particles made of a magnetic material, such as a magnetite. So a key feature of ferrofluids is the magnetic polarization of its particles, or as we call it, magnetization, abbreviated as capital M, which refers to the alignment of these particles within the fluid with the external applied magnetic field H. Due to this magnetization, a magnetic torque is generated, given by M cross H, which directly affects the microrotation of the particles. Due to all of this, we need to solve an additional equation in the system, the so-called constitutive magnetization equation, which should take into the account the existing microrotation of the particles. We refer to this phenomenon as micromagnetorotation, abbreviated as MMR. So what about blood? Blood consists of plasma, which makes up about 55% of its total volume and is primarily water, and of the blood cells, which are divided to the red blood cells, the white blood cells, and the platelets. As we know, the red blood cells are responsible for transporting oxygen to the tissues. The white blood cells protect us against diseases, and the platelets enable blood clotting, preventing hemorrhages. The blood flows in our arteries, driven by a constant pressure gradient from the difference between the diastolic and the systolic pressure of our hearts. Blood can be modeled as a micropolar fluid due to the presence of the blood cells that are suspended in the plasma. Moreover, it can be modeled as a ferrofluid when it is under the influence of external applied magnetic fields due to the existence of the hemoglobin in the red blood cells, which contains an iron atom, which is a magnetic material. Based on what we have discussed so far, here is the motivation of the research. So there are many prior analytical and computational papers on micropolar blood flows under the influence of external applied magnetic fields that account for the Lorentz force, which is the force due to the magnetic field flow interaction, but they neglect the red blood cells magnetization and thus the MMR term. I need to say that there is a small influence of the Lorentz force on blood flows, mainly due to the low electrical conductivity of the blood, which means that we have no noteworthy changes in the blood's velocity and temperature. On the other hand, we have experimental studies on MHD blood flows, which have shown that we have a reduction of the Reynolds number. We will talk about this number later. In a blood flow through cylinder by 23% when the applied magnetic field was increased from 1 Tesla to 5 Tesla. And we also have seen a reduction of blood's velocity and temperature in a magnetic field of strength of 8 Tesla. You see, all the above experimental findings, all these two, show that the Lorentz force alone, without including this MMR term, cannot explain this observation. We need a new mathematical theory to model accurately and predict the effect of the magnetic fields on blood flows. So what are the objectives of the research? First, we need to study blood flows under the effect of strong magnetic fields, which is what we need to do, using the correct mathematical model that combines micropolar fluid theory, MHD phenomena, which refer to all these phenomena from the magnetic field, and the red blood cells magnetization, and thus the MMR term. First, we should study analytically the effect of the MMR term on velocity, macrorotation, and the temperature in simple two-dimensional blood flows. Then, we need to develop a numerical code for studying more complex blood flows with or without the effect of the MMR term for comparison. After that, using this code, we can study computationally blood flows in simple and pathological arteries such as the stenosis and aneurysms, with emphasis on the effect of the MMR term. The main research goal here is to define, develop, test, and apply a mathematical computational model that can accurately predict the effect of strong magnetic fields on the human cardiovascular system. So, excuse me, what are the clinical applications of this research? This research found applications on any processes that involves the application of magnetic fields on humans. Some of them are here. First, we have the MRI examination, where the increase of the applied magnetic field strength leads to higher diagnostic accuracy. Second, we have the magnetic drug delivery, which is a targeted drug delivery to organs and tissues using nanoparticles guided by a high-intensity magnetic field. Third, we have the magnetic hyperthermia, which is a targeted heating of cancerous tumors using nanoparticles and high-intensity magnetic fields. Finally, we have the thrombosys treatment through targeted magnetic drug delivery, either using nanoparticles or a ferromagnetic fluid. So here, you can see the correct mathematical model for the application of magnetic fields on blood. It is pretty complex, as you can see, so I won't dive into many details. Just to mention here that the MMR term, which is this term, in the equation of change of the internal angular momentum, directly affects the microrotation of the particles. As you can see here, it is an equation that we need to solve for finding microrotation of the particles. Moreover, as you can see here in the constitutive relation for the magnetization, this magnetization, the latter, is influenced by the cross product between the applied magnetic field and the microrotation of the particles. This means that the magnetization and the microrotation of our particles are coupled, each one affecting the other. Let's now discuss our first study for the effect of the MMR term on a 2D blood flow using analytical methods. Here you can see the flow configuration. You can see it on the right. We have the velocity, which is defined along the z-axis, while the microrotation and the vorticity, being pseudo-vectors, are defined along the y-axis. We pose a constant pressure gradient along the z-axis to resemble a realistic blood flow. We have the magnetic field applied perpendicular to the flow along the x-axis. Of course, we have both velocity and microrotation being zero at the walls of our flow. And also we have an asymmetric heating in the boundaries with the temperature at the upper plate being larger than the temperature of the lower plates. So here we have results for the effect of the MMR term on the flow for different magnetic field values. We have two magnetic field values, one at the 0.1 Tesla on the left, and at 1 Tesla on the right. In the first row, we have results for the velocity of our blood flow, and on the second row, we have the results for the microrotation. We can immediately see that as our magnetic field increases, both velocity here and microrotation are reduced due to the increase of the strength of the magnetic field, which has a general braking effect. Moreover, when we have the MMR term here in the dashed lines, we can see an additional reduction in the velocity and microrotation, which means that the MMR term is an additional braking term in our flow. So here we have results for the effect of the MMR term on the temperature, again for different magnetic field values. Again, we have two values for the applied magnetic field at 0.1 Tesla and at 1 Tesla. On the left, we have the case where the MMR term is not included. In the middle, we have the case where the MMR term is included. And on the right, we have the relative difference between the two cases. We can immediately see that we have a significant temperature reduction at about 1.8% for the 0.1 Tesla magnetic field and at 3.6% for the 1 Tesla magnetic field. This result aligns very well with the braking effect that the MMR term has on our flow. So let's move on to the second flow case, which is for the effect of the MMR term on a three-dimensional blood flow through a simple artery. Here we're using numerical solution. Again, we have the flow configuration. You can see it here, along with the mesh that I used for solving this flow case. Our blood flows through a 3D cylinder with a nominal diameter of 3 millimeters and a length of 6 centimeters, which corresponds to a small artery. Again, we have a pressure gradient imposed along the z-axis to resemble a realistic blood flow. The magnetic field is applied perpendicular to the flow. Again, both velocity and microrotation are zero at the walls of the flow. And I need to say here that this flow was solved using two in-house built-in codes, namely EPOT micropolar form and EPOT MMR form, built from me, which were tested using previous analytical results. Both these solvers are publicly left in this link here in GitHub. So anyone who wants to work with them is welcome to download these solvers from here. And also, I need to say that the hematocrit value was constant at 45%. Here we have the results of the effect of the MMR term of the flow on the flow as previously. Again, we have two magnetic field values, but here is at 1 Tesla and at 5 Tesla. We have the black lines in the graphs, abbreviated as MHD, which refer to the case where the MMR term is not included, while the red lines here, abbreviated as MMR, refer to the case where the MMR term is included. We can immediately see that we have a significant reduction in all graphs for both velocity and microrotation when the MMR term is taken into account. Moreover, I want you to focus here at the 5 Tesla magnetic field, the microrotation, here for the MMR term that was included. We have that the microrotation tends to 0, which physically means that the erythrocytes, the red blood cells, are polarized in the direction of the applied magnetic field, allowing no internal rotation. This is actually what makes the MMR term being a breaking term in our flow. So here we have results for the Reynolds number. The Reynolds number is an important number in fluid mechanics, which characterizes if our flow is laminar or turbulent. I have the results here for different magnetic field values, for different hematocrit values, and for both the case of the MHD, where the MMR term is not included, and the MMR cases where the MMR term is included. I want you to focus on the cases that the intensity of the magnetic field is increased from 1 Tesla to 5 Tesla, both at the MHD and the MMR cases. We can see that for the MHD, we have a very small, a very tiny reduction of the Reynolds number. But when the MMR term is included, we have a reduction of the Reynolds number almost by 22.1%. This result aligns very well with the experiment of Pai et al. In 1999, which predicts a 23% reduction in the same flow. This means that the mathematical models that we use and the solvers that we created work very well, and they're aligned with the experimental results. So let's now see the last flow, which refers to the effect of the MMR term on a 3D blood flow through a simple stenosis, again using numerical solution. So here we have again the flow configuration and the mass. Again, the blood flows through a 67% stenosis, 3D stenosis, with a nominal diameter of 3 millimeters and a length of 3 centimeters. Again, we have a pressure gradient imposed along the z-axis. The magnetic field is applied perpendicular to the flow. The velocity and microrotation are 0 at the walls. We used EPOT micropolar foam and EPOT MMR foam for the solution of this flow, and the hematocrit value was constant at 45%. Here are last results, which refer to the effect of the MMR term on the streamlines of the stenosis. I want you to focus first on this kind of flow, which is the flow where no magnetic field is applied. We can see that our velocity is decreased inside the stenosis, reaching its maximum value at the point where the stenosis is most tight. Then it reaches to its initial inlet value at the end of the stenosis. Moreover, we can see the formation of these two vortices, which is a very typical result for this kind of flows. When we apply a magnetic field of 1 Tesla on the flow and the MMR term is ignored, we do not observe any significant changes between this flow here and the non-magnetic flow case here. But for the same applied magnetic field of 1 Tesla, when the MMR term is included, we do observe some significant changes with a velocity reduction up to 25% and a significant vortex damping at the end of the stenosis. These results here align very well with the breaking nature of the MMR term that we saw in the previous results. So in conclusion, what did we see? We saw significant reduction in velocity, microrotation, and temperature in a 2D blood flow when the MMR term was included. We saw significant reduction in velocity and microrotation in a blood flow through a simple artery because of the effect of the MMR term, which physically was explained as the alignment of the red blood cells with the applied magnetic field. We saw a reduction of the Reynolds number by 22.1% when the magnetic field was increased from 1 Tesla to 5 Tesla. And this result was consistent with the experiment of Pai et al., which predicted a 23% reduction. Finally, we saw significant vortex damping and notable decrease in velocity in blood flow through a stenosis due to the effect of the MMR term. Of course, the key takeaway of this presentation is that all the above results cannot be solely explained by the effect of the Lorentz force on blood alone. It's the inclusion of the MMR term that allows us for more accurate modeling and prediction of the magnetic field effect on blood flows. So how can this research be extended? We can study the effect of the MMR term on the heat transfer in complex blood flow geometries, which can have applications in magnetic hyperthermia. We can study the effect of the MMR term on turbulent flow in large arteries. And finally, we can study the effect of the MMR term using different constitutive magnetization equations. Finally, the long-term goal of this research is to provide a valuable tool for clinicians and industry in applications involving the use of magnetic fields on humans. So thank you very much. And if you have any questions, I'm very happy to answer them.